Loading. Accessor.
At the beginning of the project, the cost to discover just one base in the 3 billion base human DNA sequence was about $10. Building on this technical foundation, the automation technologies and techniques improved dramatically until by the end of the project in 2003, the cost to sequence a single base had decreased 100-fold to less than 10 cents a base. At roughly the same time, Dr. Smith and his colleagues around the world were working under the federally financed Human Genome Project. Another privately funded company, Celera Genomics in Maryland, was also pursuing the same goal using a different automated strategy called shotgun sequencing. Well, both strategies worked, and both achieved their final goal at almost exactly the same time in 2003. By then, both also had an error rate of only one base in every 100,000 base pairs. That's 10 times more accurate than they had originally predicted. The big surprise, however, was that, contrary to almost all expectations, both groups found many, many fewer human genes than just about all geneticists had predicted. It seems clear now that human beings are constructed with a blueprint of only 20,000 to 25,000 working genes, rather than the 100,000 that had been predicted just a few years ago. Well, up until now, we have shown only the basic research. What good will it do?
If you want to understand biology at a really comprehensive level or systematic level, you can kind of pretty quickly imagine that you'd want, at a minimum, to have the information about, what is the biology made of, right? It's hard to analyze something if you don't know what you're analyzing. And basically the sequence of all the genes that encodes the structure of all the proteins is sort of like the minimal fundamental building block that you can use. And although there's a lot of resistance to this concept when the Genome Project started, that resistance has pretty much gone away, in part because the power and utility of this information has become so obvious that nobody can overlook it. So for instance, the yeast genome is now completely sequenced. It's about 10 million bases. And yeast biologists all over the world are all using the yeast genome information all the time. And even yeast, which you might say is relatively simple, 10 million base genome instead of 3 billion, and a unicellular organism instead of a complex organism like us, even there we know remarkably little about, even with all this sequence still, about what really makes this thing tick. How does it divide? How does it control its growth? How does it respond to its environment? How does it shuttle and move and control its gene expression? All of those questions are big, hard questions, which work's going to go on for a long time. And what you do when you produce all this sequence is you give people a very powerful sort of infrastructure tool to help them go after those hard questions.
Scientists now are also rapidly sequencing the genomes of many other species. Besides yeast, the genomic sequences of bacteria, roundworms, mustard plants, fruit flies, mice, monkeys, chimpanzees, as well as many other species of plants and animals have already been sequenced. Many gene sequences are the same in species as different as yeast and human beings, showing once again the basic unity of all living things on this earth. As more and more genomes become sequenced and available to researchers, we will have a more and more complete understanding of the evolution of all plant and animal species on Earth, including our own species, Homo sapiens. And by comparing DNA sequences in humans from around the world, we can already begin to trace a clearer line of evolutionary change and development from our ancestors in Africa to the many ethnic, racial, and even down to the individuals in the world today. So far, we have emphasized the likeness of DNA in all species of living things and talked of human DNA as though it was the same in all humans. Well, it is over 99.9% the same in all humans, but that is not exactly the same. In fact, except for identical twins, each human being has a unique genome. Remember, human chromosomes come in pairs. One chromosome from your mother and one from your father. That means the genes come in pairs too. One set from your mother, one set from your father. In order to make the protein hemoglobin, for instance, you have not one but two genes. If both these genes are exactly the same normal code, there's no problem. You have normal hemoglobin and normal red blood cells. However, sometimes a gene for making this protein can have a mistake in the base code. What is called a mutation happened sometime in the past. That is, the normal gene changed. Maybe just one base in the long sequence got knocked out of order somehow. It's called a SNP. This mutated gene, this SNP, gets passed on to children. In the case of the hemoglobin gene, an unfortunately fairly common mutation can lead to a serious hereditary disease called sickle cell anemia. In sickle cell anemia, the red blood cells take on a sickle shape and cannot as easily carry oxygen to body cells. Now, if one of the hemoglobin gene pair is normal and the other member of the pair is defective, the person will not have the disease. However, this person will be a carrier of the mutated gene. When he or she has a child, the child has a 50-50 chance of getting this mutated gene. Well, if both parents are carriers and each passes on the mutated gene, in the child both hemoglobin genes in the pair would be defective, and the child would have the disease. We think of mutated genes as bad genes, and they usually are. In the long run of evolution, however, human beings are the result of mutated genes that were good. In fact, every one of our 20,000 to 25,000 genes is a successful mutant gene, created and then passed on over 4 billion years by the evolutionary process. And in fact, it is these mutant genes that make us all delightfully different from each other. It is mutant genes, in other words, that have led to the incredible diversity of living things on this living earth. In the short run, though, mutant genes are often a serious problem in the human species. Diseases like sickle cell anemia, cystic fibrosis, Tay-Sachs, Down syndrome, and Huntington disease are all caused by a mutation in a single gene. Now, here is one place where the Human Genome Project may be of great help in the future. Dr. Smith explains just one example of benefits already realized from basic research in the human genome.
There's some good examples of that, actually. Another company I work with called Visible Genetics has been doing sequencing of the retinoblastoma gene. So it's a gene that's involved in generation of cancers of the eye. And it turns out that if you're in a family that has that gene, then and if you don't do any genetic testing, then you don't know which of your kids have those genes and which don't. And it turns out, since you don't know, what they end up having to do is they have to do these examinations under general anesthesia of the eye, which are pretty expensive tests, and they have to do them like every 6 months on young children to detect if there's going to be an early occurrence of this eye cancer, so they can then try to ablate it by laser surgery at an early stage. So it's a very expensive test, and it's very stressful for the children who are exposed to it, and it's stressful for the whole family, and everybody's sort of sitting on a bomb and they don't know when it's going to go off. So Visible Genetics developed this test that allows them to go in and rapidly sequence those genes from the affected members in the family. Once they do that, they can find out what the mutation is in the gene that's causing the problem, and that allows them to then go and very quickly and easily test the children and find out which children have the bad gene and which don't. The ones that don't, right away are free and clear. Okay, they're out of it. No test, no general anesthesia, no anxiety. And so that's a huge benefit right away. And then the ones that do, you can also start building up a database to look at what the prognosis is based on different types of mutations, and also try to tailor treatments that are specific for those mutations. So in general, that's just one interesting example of the social payoff for this. And the social payoff is really going towards targeted treatment for people based on their genetic type, their genotype.
As in all other fields of science and technology, when genuine knowledge increases, there are benefits and there are risks. Automobiles in the 20th century changed the world by making it easier for humans to move around. Few people would want to give up the benefits. On the other hand, more people have been maimed and killed in automobile crashes than in all the wars of the 20th century. So too, the Human Genome Project will dramatically increase our ability to diagnose, to cure, and to prevent many diseases. Some critics worry that it will also give us more information than we want to know. Let's say a doctor finds in a routine physical that would include a genetic scan. He finds out that you carry a gene that predicts with high probability you getting breast cancer, or prostate cancer, or heart attack at an early age. You may or you may not want to know this, but you almost certainly do not want your insurance company to know it. In the future, will insurance companies require this kind of genetic testing as a prerequisite for giving you insurance? Well, legislatures around the country are already considering laws to prevent this from happening. Genetic studies have already shown that at least 5,000 or 6,000 human genes out of the 20,000 to 25,000 total do have mutations that can cause health problems. Some minor, others very serious. Example of the latter would be cystic fibrosis, Huntington's disease, diabetes, muscular dystrophy, and perhaps Alzheimer's disease and some forms of cancer. As with sickle cell anemia, many people are carriers of these genes, but do not actually have the disease because the gene on the opposite chromosome in the pair is normal. Well, knowledge gained in the Genome Project will enable technicians to test and discover whether you carry a mutated gene in your unique genome. What do you do when you find out that you do carry this gene and your spouse also carries it? In that case, there's a 25% chance any children you have will inherit the disease. Now, here's one possibility that has already been used. Using new in vitro fertilization techniques, doctors can genetically screen a number of very early embryos while they were still in a culture dish and find out whether they have the gene pair that produces the disease. Then only embryos that do not carry the mutated gene pair will be implanted in the mother's uterus. Of course, using the same technique some critics have pointed out, couples could in the more distant future not only prevent serious diseases from being passed on, they could also choose among desirable genes for their offspring. While there is no single gene that leads to good looks, or intelligence, or athletic ability, or musical genius, there is a strong genetic component to these traits. But when the complete human genome is worked out, we will probably know more about which genes are involved in these complex human traits. Armed with this knowledge, you might be able to screen embryos for genes that would lead to children with not only no genetic defects, but for children with desired sex, height, body build, athletic talent, musical talent, mathematical talent, and so forth and so forth. Some have even envisioned the day will come when prospective parents could sit before a computer screen and choose among many alternatives what traits they want their next child to have. It's not inconceivable that cut-and-paste techniques already developed in other plant and animal genetics could then be used to insert desirable genes or remove undesirable ones. There is even the possibility of adding an entire chromosome with many novel genes to the embryo, genes never before a part of the human genome. Other animals, for instance, have genes for seeing infrared or ultraviolet light. You might be able to insert a gene for such a capability into a human embryo. Or you could insert a gene to enable the offspring to detect odors as well as a dog or a cat. Or to navigate by sonar like a bat. Or generate electricity like an eel. Or navigate magnetically like a bird. Or, well, Leo M. Silver, a professor of molecular biology at Princeton University, has gone so far as to predict that such genetic screening and reconstruction is not only possible, but inevitable. He thinks that by the 22nd or 23rd century, this may well be the norm. That is, for educated, well-to-do parents who can afford the expense. What about parents who either cannot afford the expense or decide on religious or moral grounds they do not want to interfere with nature in these dramatic new ways? Well, the long-term effect of this split may be surprising, says Silver. Controlling human genetic continuity for the first time in history may lead to a truly revolutionary change in a few centuries, a blink of the eyelash when it comes to evolution. The well-to-do, scientifically oriented humans, say Silver, may well make themselves into a different subspecies from natural humans. And in fact, human-controlled evolution may accelerate to the point where there will be at least two varieties of Homo sapiens, so distinct in their genomes that they will be actually unable to mate and produce fertile offspring. Silver calls his book Remaking Eden to call attention to the revolutionary events that may accompany that step into the genetic unknown. Whatever actually happens in the 21st century, it seems certain that the words of Neil Armstrong, when he first landed on the moon, will apply even more aptly to the Human Genome Project. A small step for man, a giant step for mankind.
New notification. DualSense wireless controller connected. Battery charging. Low battery.
Chemists, physicists, biologists, and computer scientists from around the world have now completed the most ambitious biological research project of all time, the Human Genome Project. One of the lead scientists who worked overtime on this project was Dr. Lloyd Smith, a chemist at the University of Wisconsin in Madison. We asked him just what is the